So thank you so much. It's a pleasure to be here. Thanks, Max, for inviting and organizing. It's really great to be here. I'm not a physicist, so I'm not sure if I'm in the right audience, but I will try. So my lab tries to understand the phenotypes that are associated with aging. So what's driving the molecular changes that we see that, for example, drives graying of hair or facial wrinkles or changes in fat distribution or pigmentation changes, and why do we, in essence, why do we eventually die, right? And so there are many, many phenotypes, more than what you can see on the surface. This is a paper where we downloaded PubMed, and then we looked at which features are associated with aging, and then we correlated that with actual cohort data. And so graying of hair is actually the most prevalent feature if you look at a population, so 99% of everybody that's old will develop graying of hair. So in a sense, you can say that's a pretty good biomarker of aging. If you go to Wikipedia, though, I just learned this, graying of hair is not a biomarker of aging, according to Wikipedia. But I would argue that it's actually a quite good potential biomarker of aging. But there are other things that occur, other phenotypes associated with aging that are much more serious, of course. So cancer is something that will occur in about 40% of everybody, and then there are many things, right? And this also indicates that we're really phenotypically quite diverse. The older we get, the more phenotypically diverse we are. And so another way that you can look at this is you can look at phenotypes of other diseases. So this is a disease where you have issues with your telomeres. And so these are patients that have single gene mutations that lead to loss of telomerase, and that leads to some features that could be age-associated, so skin pigmentation changes are age-associated, but sort of classic progeroid features are actually not seen if you have issues with your telomeres. So, and there's a wide variety of these types of genetic diseases that we can then use to understand what occurs with age. And so you can, and this is, I've included this because there's some equations here. Please don't ask me to explain them. But this is basically just hierarchical clustering, right? So it's a correlation coefficient, and then how to determine linkage. And so basically what you can see here is that each column is a disease, and then each row is a phenotype. And so you can then look at overlapping phenotypes to try to understand which phenotypes are co-associated, so which phenotypes occur in the same diseases, suggesting that those genes where you see those phenotypes could be linked to the same pathways. And so I'm going to give you some unpublished data on one disease that we identified. We used this aging phenome, so this collection of these different phenotypes associated with aging. And then we went to the online Mendelian inheritance in man database to identify diseases that were associated or that showed overlap with aging. So the number of shared features and the weights. So if you have graying of hair, that has a stronger weight than, for example, Parkinsonism, which is age-associated but occurs at a much lower rate in aging individuals. And so Wood-Sakati syndrome is a disorder that is caused by single gene mutation. It's very rare. There's a few of these patients described. And looking at the phenotypical similarities, there's actually quite strong overlap with known premature aging disease. So it clusters with aging. We looked for whether or not patients that suffered from Wood-Sakati syndrome, whether or not they displayed accelerated aging. Here we used a biomarker that sometimes worked, the face. So we looked at facial photographs and then the age prediction on facial photographs. So the blue dots here are Hodgkin-Gilford progeria patients. They're known to be prematurely aged. Werner syndrome are yellow. They are much slower progressing premature aging disorder, but they are also prematurely aged. And these patients also actually display premature aging in the face, at least according to this AI algorithm. we also looked using, so we identified the first family actually in Northern Europe that suffers from this disease, and then we found a couple of, we got some blood samples from these patients, and we did the aging AI algorithm, which is similar to this blood biochemistry algorithm that's been developed here in Singapore. And we saw that patients showed accelerated aging in that clot. So we mutated the gene that's responsible for the disease called DCAF17, and we expressed DCAF17 in cells and identified it to be recruited to sites of DNA damage. So these are cells where we are shooting them with a laser, and then they get recruited. We identified the interactome so the proteins that are interacting with this gene DCAF17. So this is this DDB1 called 4 complex, which is involved in ubiquitination of other proteins. So it's involved in protein stability and identifying substrates for where this needs to go. And so we can identify a single amino acid that's important for this. If we mutate this amino acid, it doesn't get recruited to DNA damage sites anymore. So knockout mice, and this is why I mentioned to Matt, what about the short-lived control? So in knockout mice, the mice are sterile. They have inflammation in the testes, and they have increased frailty when doing frailty scoring, and they are shorter lived, the knockout mice, compared to wild type, but the wild type are already quite short-lived, so I'm not sure how to conclude that in terms of Matt's thoughts. We also identified a target for this complex, one of them THRAP3, which is a gene that's been involved in R-loop resolution. And when you damage cells, so this is a cell, this is THRAP3, when you induce DNA damage, you can see with this gamma H2AX, you can actually see that THRAP3 is excluded from the DNA damage site. If you lack DCAF17, you get stronger exclusion. So it suggests that DCAF17 stabilizes THRAP3 at the DNA damage site. And indeed, if we can see there's less THRAP3 in the testes of these mice. And THRAP3 has been involved in R-loop resolution. So you knock down DCAF17, you get more R-loops. You also get more single-stranded DNA. There's higher amount of RPA, particularly in the chromatin fraction. And when we did DRIP-seq, we see more R-loops, in particular much more variation in R-loops. So it seems that the regulation of R-loops is lost when you lose DCAF17. So the idea is that if you don't have DCAF17, you get R-loop instability, and then that can lead to premature aging. So this came from one phenotype where we looked at, or one investigation where we looked at phenotypes of aging and identified a potential disease. We're also looking at phenotypes in another way. So this is data from healthcare records this is in ancient Egypt. We're not looking at this, but we're looking at pathology records in Denmark. So we have 33 million pathology reports. And we can then look at patterns of aging in these reports. So in this particular project, we basically kind of made a bag of words type of investigation where we generate a very large spreadsheet where we have the date of birth of an individual and the date of death of an individual, and then whatever is mentioned in that pathology report. And we can do a UMAP analysis of terms and then map it to the average age of that term, and then we can see that some terms are associated more towards the center, and then terms kind of dissociate to all directions, so suggesting that it's kind of a chaotic process, which is maybe not surprising. If you do the mean, if you take the average of each age and then do a PCA on the data, you actually get very clean data. So here is year 0, and this is year 100. And you can see that the data kind of wanders through this PCA plot and hits sort of a low point at around 20 and then wanders up towards the end of life here along this PC2. This plot here is the speed of change, so the distance between each dot. So there's a lot happens early on, and then that kind of slows down, and then around 50 there's another peak where stuff happens, and then towards the end of life there's a considerable peak also. And so we thought this peak here may be menopause related, and so we separated by female and male. And females, you can see this slope here, that, or this pattern here, that you have a low point here and then you get aging occurring along this PC2. The low point is actually around 19 years of age, where you see this sort of progression. But for males, you actually also see this kind of peak around 50. And for males, you can see that not a lot happens until around 40. Something does happen here, but it's sort of stable from 20 to 40. And then once you hit 40, it's really very fast downhill for men, unfortunately. Another thing is that you can see the principal component 1 is actually, so it's aging seems to be a combination of PC1 and PC2 in this plot, and it's really describing a large majority of the variation in the data set. So aging really has a very strong effect there. We can also look at individual tissues and look at how terms associated with age, but you can also look at, because we have death registry, we can also look at sort of do Cox regression on individual terms and see hazard ratios for individual terms. And then we can also take these terms, identify terms that are associated with slower aging. In this context, it's slower lung aging, and then we have our PubMed data set, and then we can look for other things that are associated with slower aging. In this case, we looked at lung aging and identified a drug, nintedanib. We didn't identify it. It's, of course, identified before, also in the context of lung, and we saw that it reduces some aspect of cellular senescence, and in fruit flies, at least, it increases the lifespan of the fruit flies. So this is sort of the overall idea, that you can go from like what does a pathologist see in the microscope and what he writes down, and then eventually to identify molecules. And I just bring this because this is based on registry data. We have very good registry data in Denmark, and this is, I think, one of the largest registry data sets perhaps in the world, which is looking, we have registry data on more than 1 billion prescriptions. So basically the drugs that individuals are taking, and we can correlate that with diagnoses, and we can correlate that with the lifespan. And we can also then look for combinations as Matt indicated. That would be really interesting to do this. 3,500 FDA-approved drugs in this database. And so it's a very interesting data set. So another thing that we're looking at are tissue phenotypes. So we have previously shown that we can identify senescent cells based on nuclear morphology. And so in our more recent work, we tried to go into healthy biopsies and see if we can identify if nuclear morphology in these biopsies can predict health outcomes, in particular cancer. And so we can identify changes in different in nuclear morphometrics between different tissues. So we can identify stromal tissues, fat tissue, epithelial tissues, and terminal ductile lobular units. And we can then go in and identify senescence in each of these tissues. And so this gives us something like this, that you can see aspects of different types of senescence in different tissue compartments, and then you can look at spatial orientation of these senescent cells. And what's interesting is that this is sort of an old finding, that senescent cells appear to cluster together, that senescence can lead to induction of senescence in other tissues. I think what's also interesting is actually that you have kind of young cells, or young non-senescent cells, that appear to also associate with other younger cells. And they're actually clinically relevant. So we can identify these cells and then look for risk of developing cancer in the future. And using various predictors, we can combine that with the Gale score and actually get a odds ratio of 4.7 in our ability to predict the risk of developing future cancer based on a biopsy from a healthy woman. And so this has implications because every year there's more than 1 million biopsies that are taken as part of mammograms in the US alone, and 80% of those are benign, so they're not cancerous. But now we can potentially actually risk stratify these. And to look more into this, we did this actually in women that have gotten tumors identified on mammograms, and then we did H&E staining, or this was done by Tom Rohan at Albert Einstein. They had done H&E staining, and we can then do risk stratification. We can risk stratify these women that have benign breast disease per definition because they have a tumor, and we can risk stratify these women now. Okay, what else can we do with this tool? So I'm just going to talk to you about two sort of use cases that we've done in the lab. One of them is looking at hypoxia. And so I have an Ethiopian PhD student in my group, and Ethiopia is a quite interesting country in terms of hypoxia because there are people that are living at very high altitudes and people that are living very low altitudes. So there's a very and there's a lot. I think it's the population in the world with the most people living at high altitudes. So that means that we can look for chronic hypoxia, for example. How does that impact aging? And what's really interesting is that the higher altitude, the longer the lifespan or life expectancy, and the lower the amount of DALY. So that means that the less sick you actually are. And so this guy he's from the Tigray region of Ethiopia, so we wanted to do a deeper understanding of maybe what could contribute to this. And so we did two kind of potential markers of aging. So we looked at face aging, so we took photographs of the individuals and then looked at age prediction based on facial photographs, and then we can also do blood smears. So in Ethiopia, they're quite good at blood smears because they have malaria, or they have a high malaria burden. And so that means that this is a very low kind of tech way to get nucleated cells from a person, and then we can do senescence prediction based on the nucleus. And so what we can see is that with elevation, you get an increased predicted photo age. Maybe that's not surprising because high altitudes you get more UV exposure, but you get actually with elevation a decreased risk in DNA damage predicted senescence looking at IR and a model trained on doxorubicin, so two molecules that leads to DNA damage. And so the second I want to talk to you about is a trial on COPD. So this is smoker's lung that is caused by smoking. Smoking leads to induced DNA damage. This can lead to PARP activation. This is not a review from me, but other people think so. So if you have PARP activation, that can lead to loss of NAD. And so I've done a lot of work on this in the past. So in these premature aging disorders, PARP activation may be driving NAD decline and then leads to loss of metabolites. So we can use NR, as we heard, to maybe negate some of this. 2 seconds, then I'll be done. We did a double-blinded RCT with NR and placebo, and our primary outcome measure was IL-8, which we see is significantly reduced with NR compared to placebo. I think we also looked at senescence in the cells in the lung. I don't have time to talk to you about that, but I think this is an interesting result that COPD patients do seem to have accelerated aging. This is in blood samples. We did not see an effect on NR on these DNA methylation biomarkers of aging. What we do see, however, is that DNA methylation biomarkers of aging correlate with inflammatory markers in the blood, but also with senescence prediction in the lung. So this is just the take-home message. We have aging biomarkers, they're clinically relevant, and we can actually do interventions to influence meaningful biomarkers in humans. And with that, I'll just say thank you.
So we're running really late, so one or two quick questions.
Hey, that was really cool. I just had a quick question about the senescence prediction. Have you been able to take the ones that you predict would get cancer and split them into two groups, so they had senescence but didn't get cancer, and then figure out why some did and didn't within the ones you predicted would? Because that might then lead to some cool findings.
We're not actually, so we're measuring the hazard ratio, right?
Yeah.
So that's an interesting idea. We haven't done that, but that would be quite interesting.
Cool, thank you.
To look at.
Really quickly.
I really liked the work you did on health records and identifying this new or this genetic disease that might have some aging features. What's your sense for the phenotypic space there? Like, how did you find a lot of hits for diseases that have some overlap with aging that like haven't been characterized before?
Yeah, I mean, so there's we screen more than 5,000 more than, I think there are 5,000 genetic diseases in the database, right? And some of them are known diseases where the pathogenesis is really well known. I think there's a lot of diseases that are historically not sort of considered premature aging diseases, because typical premature aging diseases have been characterized by a physician, where the patients have gone to the physician, and then the physician looks at the patient and thinks, you look kind of old. And then he has written that down and sent it to a paper, right? And then that's how you get diagnosed with premature aging. But obviously, we age in very different ways, right? And our internal organs may not age in the same way as our skin, for example. And we know that from these premature aging diseases where you see skin aging, they often do not have aging of other tissues. So I think we also have to think about this. We're treating aging, right? But aging is actually very tissue dependent, and I think particularly dependent on whether or not tissue is replicating or not. This, I think, is a very, very strong, has a strong impact. And so here, for example, the inborn errors of metabolism, many of them are mitochondrial diseases, but mitochondrial diseases, these patients do not look prematurely aged. So when you see a mitochondrial patient, they don't have sort of classic premature aging. But if you look at overall overlap with what we would consider features associated with aging, there is an overlap. So these are different things, right? Aging is more than skin deep.
Thanks.
Thanks, Morten.